right, let's jump right in. Today, we're going on a journey from the medicine cabinet you know, full of simple pills, to the absolute cutting edge, living, programmable medicine. We're going to break down how biologics, biosimilars, and even our very own cells are being turned into powerful new therapies. And look, this isn't just some cool science story. It's a revolution that demands a whole new playbook for healthcare professionals, especially the pharmacists who are right on the front lines, making sure these advanced treatments get to the people who need them.
Okay, so to really get this, let's start with a basic question. What's the real difference between two very different kinds of drugs that have completely rewritten the rules of medicine?
So on one side, you have the pills we're all familiar with. In the biz, they're called small molecules. Why? Because they're simple, they're cooked up from chemicals in a lab, and they have a really well-defined structure. But then you've got biologics. These things are totally different. They're massive, complex proteins that are actually manufactured inside living cells. And trust me, that difference isn't just a science class detail. It changes absolutely everything.
And what's so fascinating here is how these fundamental differences completely change how the drugs behave in your body. Small molecules, you swallow them as a pill, your liver breaks them down, and they don't last very long. Biologics, on the other hand, are way too big and fragile to be taken as a pill. Your stomach would just destroy them, so they have to be injected. But the upside of their huge size is that they can hang around in your body for weeks, which means you don't need to take them as often. See, it's not just a different drug, it's a totally different philosophy of treatment.
Okay, so let's build on that and take it to the next level with one of the most powerful types of biologics out there, monoclonal antibodies. You'll often just hear them called mAbs.
You know, the best way to think about these is as heat-seeking missiles for your immune system. Seriously. They are engineered in a lab to find and lock on to one single unique target, say a specific protein that's only on the surface of a cancer cell. And they do it with incredible precision, completely ignoring all the healthy cells around it.
And this just brilliantly shows one of their main strategies. It's like a three-step special ops mission. First, the antibody finds the bad guy, the target cell, and tags it. Second, the other end of the antibody acts like a bright, flashing beacon, calling in the immune system's heavy hitters, cells like natural killer cells. And finally, those recruited cells show up and destroy the tagged target. It's a process called ADCC, and it's basically technology supercharging what our bodies are already designed to do.
But it's not just about antibodies that tag and destroy. The biologic toolkit has all sorts of clever tools. For instance, in diseases like rheumatoid arthritis, you've got all these harmful signals floating around that cause inflammation and damage. So clever scientists design fusion proteins, like etanercept, to basically act like a molecular sponge. They just float around, soak up, and neutralize those bad signals before they can cause any trouble.
Now, these revolutionary medicines are amazing, but they are also incredibly expensive. And that's created a huge problem with access, which brings us to our next big topic, biosimilars. And with them, a really fascinating human challenge that healthcare leaders have to get right.
So what on earth is a biosimilar? The easiest way to think about it is as a highly similar version of an original biologic drug. It has to go through tons of rigorous testing to prove to regulators like the FDA that there are no clinically meaningful differences. That's the key phrase. It means it's just as safe, just as pure, and just as effective. But here's the kicker. It opens the door to competition, which helps bring those sky-high costs back down to earth.
But here's where it gets kind of weird and very human. We've all heard of the placebo effect, right? Where your belief that something will work can actually make it work better. Well, the dark twin of that is called the nocebo effect. It comes from the Latin for I shall harm. And it's a powerful, real phenomenon where if a patient expects a medicine to be worse or has negative feelings about it, they can actually experience real negative side effects.
So this is absolutely crucial for frontline clinicians like pharmacists. Switching a patient to a biosimilar is about so much more than just changing a prescription. The communication itself becomes a clinical intervention. You need a phased, empathetic strategy. You have to positively frame the change, listen to and validate a patient's concerns. This isn't a soft skill, it's an essential clinical tool to prevent that nocebo effect and make sure the therapy actually works.
Okay, everybody take a deep breath because we are about to take a massive leap. We're moving beyond manufacturing drugs in labs and into the ultimate form of personalized medicine, literally programming our own cells to become the cure. Welcome to the world of cell and gene therapy, or CGT.
Broadly speaking, there are two ways to do this. The in vivo approach is like sending in a microscopic delivery drone, usually a harmless virus that's been retooled,
directly into the body to drop off a new, healthy gene in a specific organ. Then there's the ex vivo approach. This is more like taking your cells out of your body, sending them to a super high-tech repair shop where they get upgraded, and then infusing them back in as a living drug. And that ex vivo idea is perfectly captured by something called CAR-T therapy. And wow, is it an incredible journey. You start by collecting a patient's own immune T cells. Those cells are then shipped off to a specialized lab where they get reprogrammed to become expert cancer hunters. Meanwhile, the patient gets chemotherapy to clear out some space in their immune system. And then finally, their own supercharged cells are infused back into their body. It is an unbelievably complex process, and specialized pharmacists are absolutely critical members of the team that manages this incredible living medicine.
And please, do not for a second think this is just science fiction. This is reality, right now. Zolgensma can stop a devastating disease in its tracks with a single infusion. Kymriah is curing leukemias that used to be a death sentence. And Casgevy, the very first approved CRISPR therapy, is offering a potential cure for sickle cell disease by literally editing a patient's DNA. These are real medicines changing real lives.
So, if these therapies are so incredibly powerful and personalized, how do we make sure they're not just effective, but also safe for every single unique individual? Well, the answer is already written inside of us, in our DNA.
There's a whole field dedicated to this called pharmacogenomics, or PGX for short. It's a fancy name, but the idea is simple. It's about using a patient's genetic blueprint to predict exactly how they will react to a specific drug. It's the absolute key to getting away from one-size-fits-all medicine and moving towards giving the right drug at the right dose to the right person every single time. And in some cases, a genetic test isn't just a good idea, it is absolutely mandatory. For the HIV drug abacavir, you have to get a genetic test first because if you have a certain marker, you're almost guaranteed to have a life-threatening allergic reaction. For the cancer drug trastuzumab, you have to test for the HER2 protein because if the cancer doesn't have it, the multi-million-dollar drug simply will not work. This is the future, right here, using our own genetic code to maximize good outcomes and eliminate harm before a single dose is ever given.
So let's just take a step back and think about the ground we've covered. We went from a simple chemical pill to a precisely targeted protein all the way to a living programmed cell. The very definition of what a medicine is has been completely transformed. The playbook is rewritten, and it demands a whole new level of expertise in science, in communication, and in logistics from the clinicians who are wielding these powerful new tools. And that leaves us with one final big question to think about. As medicine evolves into a living technology, are we truly ready for the immense responsibility that comes with it?